Good morning, everyone, and thank you very much for this opportunity to present some of our results associated with this Cawthron report 2980, which was supported by a range of stakeholders, including MDC, and which aim was to develop a novel molecular-based index for rapid and cost-effective assessment of fish farm impact on the seabed biology. So before I go too much into technical details, I will give you a little bit of background on the use of traditional versus more novel monitoring techniques, and as well as a little bit of contextual historical context from where these data came about. So, traditional biomonitoring of fish farm impact currently involves the collection of large amount of sediments, which are collected along enrichment gradients from unenriched sites far away from the farms and along transects all the way to the fish cages. And in this process, a number of biological and chemical data are actually collected. The biological data typically involve the use of microscopes, which to identify and count the thousands of organisms that live in the seabed. And these data are then combined into the so-called enrichment stage index, which you are all familiar here, which ranges between 1 or no impact, very unenriched condition, to highly enriched conditions, or 7, and the level of 5 being the upper regulatory limit here in the Marlborough Sounds. So this is the gold standard. It works really, really well, but it has a number of clear limitations, mostly time and cost. So if I drive you through the workflow of these traditional methods a bit more, it takes typically about 1 to 3 days to design any project. To that, depending on the number of fish farms, it will take you at least a week to collect the samples. The major time bottleneck is at the microscopic stage, at the analysis of the macroinvertebrates. To that, you can add at least a month in data interpretation results, generating a report, and a few more days to inform the client. And this is only when you can finally take action. And if you sum all of that, that takes 4 to 5 months. This is too long for allowing regulators and the fish farm managers to take appropriate actions, because a lot of things can happen in 5 months. So it is a labor-intensive technique. It is also expensive, costs about $350 one sample for just the taxonomic analysis. And so there is a clear need for more rapid and cost-effective techniques. So in recent years, the last few years, there's been a real revolution in the field of molecular biology with incredible new technologies that came about. One of them is nicknamed as EDNA Metabarcoding. It looks at barcodes in the DNA, which involve the current workflow. So briefly, it involves the collection of very small amount of sediment. So we're talking grams versus kilograms with traditional techniques, and from which all of the DNA or the genetic material is extracted with all of the traces of organisms in them. And then we're using this so-called high-throughput sequencing technology, which allows to sequence in mass all of the little fragments, genetic fragments, that allows us to identify organisms, all of the organisms that are found in them. Then we use quite a lot of bioinformatics process, which clean the data, sorts them, organize them, and then allows you to compare those data to reference sequence database, which allow us to identify organisms like a fish here or whatever along, all across all of the organisms. And of course, the output files are very similar to those used in traditional ecology for analyzing richness or biotic indices. There are a number of clear, significant advantages of this technique over traditional. It allows much faster turnaround, reduced cost, uses very small sample size, which are easier to obtain in the field. It has a greater sensitivity and specificity. It can also look at the entire tree of life rather than just focusing on a narrow group of organisms. And very importantly, it can be used very effectively to standardize the approach so that different laboratories around the country and around the world use the same workflow to get the same answer at the end. And of course, it is very good to target very difficult environments, which traditional techniques sometimes cannot explore. So, in a proof of concept back in 2012, we were among the first in the world scientists to actually apply this technology to the monitoring of fish farms. And this was supported by the Ministry of Business, Innovation and Employment at the time, and we focused on a very specific group of organisms. These are microscopic organisms called Foraminifera, which are very abundant in all sediments and which are good bioindicators of pollution. Through this work, we demonstrated that yes, the Foraminifera can provide some excellent bioindicators, and we also started the base of generating a new molecular-based index that you can see here, and confronted it to the traditional enrichment stage, and it performed pretty well. These were published in the international literature and Cawthron Report 2734. We went then further than that and started to look at other organisms, and in this case, bacterial communities. Bacterials are amazing. They turned out to be even better indicators of rapid environmental change, both in fish farms as well as under mussel farms in the fields of Thames. Sorry, my French accent. And this is reported in Cawthron 2880. So, today, this project here explored the ability and the robustness of metabarcoding, this new technique, in developing a new index of fish farm impacts, but with the end goal of reducing monitoring costs and reporting times. The real stretch or novelty of this approach was really that for the first time, we looked at three large taxonomic groups. We looked at all bacteria, all foraminifera, and all eukaryotes. So eukaryotes are essentially all protists, animals, and plants. So we essentially look across the tree of life and incorporate multiple trophic level. So this is a world first. And then also, first time is that we looked into incorporating samples from multiple years and different farms, high flow and low flow. So that was also novel. The two main questions were, first, does the community composition within each of these three taxonomic groups changes consistently across enrichment stage of fish farms in space and time? And the second question was, can we actually develop a multi-trophic biotic index, or MBI, to apply? So we focused on three distinct farms, two farms that you are very well aware of, here in the Marlborough Sounds, low-flow farms, Otanera, and high-flow site, Tepangu. And these were surveyed over 3 consecutive years, from 2012 to 2014. And another external farm in Stewart Island in Big Glory Bay, which was collected in 2013. Now, importantly, the Biotic index was actually developed on the first 2 years. This was our assignment data set. And the third year, 2014, was used as a validation data set to test the index. The process was essentially collecting all the sediments, 105 sediments, and we processed 2 different analyses. On the one hand, the traditional approach, using microscopy to identify invertebrates and generate the enrichment stage index, and in parallel, using the same samples, we extracted all of the genetic material, used this high-throughput sequencing technology to capture all of these 3 taxonomic groups that I talked about, bioinformatic tools to compare and identify the species present within each of these groups against reference database, and of course, then a big exercise which was about developing this metabarcoding biotic index. So this slide is simply to show you the extreme power of this technology. This is only one of the 3 groups. It's the eukaryotes, so protists, animals, and plants. Across our data set, we were able to identify over 3,000 so-called operational taxonomic units, or OTUs. These approximate, in our world, it approximates the molecular species, so the species level. When you compare and confront that to fully annotated reference database, you you can obtain the diversity at different taxonomic levels, from 6 supergroups all the way down to 885 identified species. And this is represented here. This is just to say that that would have taken years using a microscope to obtain, and that was done automatically and very rapidly. Now, to address our first question, I'm going to get slightly more technical, and I apologize for the background here because it's very small, but I'll go slow. The first question was, does the community composition changes consistently across gradients in the 3 taxonomic group? So this diagram here represents the overall biodiversity or composition of one of these groups, the eukaryotes, here you see. This representation represents the community composition shown in a 2-dimensional scale. What that means is that those little icons that you see, let me point out, these two, for example, are much more similar to each other in terms of total biodiversity composition, so very similar, whereas those 2 points, for example, this one and that one here, are very distinct from each other, very different diversities, okay? Another thing is that you see different colors. So the green and the blue represents the low-flow Otanera farm, year 1 and year 2. The yellow and the orange here represent high-flow site, Tepangu, year 1 and year 2. And then the red one down there is the Big Lorry Bay site. So what you see quite a bit of a split between the low-flow sites and high-flow sites. You also see quite a bit of a split with the Big Lorry Bay. And something I forgot to say is that those numbers represent the distance in meters to the fish cage. So what that means is that on your right, you have all of the unenriched sites, and as you go to 0, this is a cage. So you see a real change in community composition from unenriched to enriched in a very linear way. Very fascinating. And the years, the community composition between years is quite stable, but there's a split between high flow and low flow. Now, just to move on to the other 2 taxonomic groups, you have here Foraminifera, and here you have the bacteria. So altogether, we can see that they all change from unenriched to enriched in the same manner, from right to left. There's also a split between low flow and high flow in the forams, but a bit of a problem here, because you see quite a lot of differences between years. So that means if you take a site, you look at forams last year, you look this year, they change quite a bit. And this is not so good for developing an index. Whereas here, the bacteria are very, very similar to each other, and regardless of year, flow, or farm. And this is really good. To address our second question, which was, can we actually develop a metabarcoding biotic index? Yes, we can, and we did. So to do that, we applied quite an in-depth analysis of all of those data sets, and we identified over 500 key ecogroups, taxa, or genetic signature which abundance consistently peaked over a given enrichment stage index that you can see down, regardless of flow, year, or farm. And we use this information to generate a series of metabarcoding biotic indices, which were then confronted using correlation analysis to the traditional index, which is found there. And we generated an index for forams, for bacteria, and for eukaryotes, and also a combined index, which included everyone, and which hopefully the assumption was that it would strengthen the solidity of the index. And it did. This is some of the results of from the assignment data sets, and what it shows, this is the combined index that is all together, shows that the metabarcoding biotic index provided over 90% strength relationship with the traditional method, which is really fantastic, absolutely excellent. Another observation was that we looked at 2 types of molecules in parallel. So from each sample, we extracted DNA and RNA. So let me just explain rapidly. DNA is the block, the forming block of life. It's a very strong molecule that is used commonly into the EDNA monitoring, but it can remain in the environment for months, for week to months. It can stay after cell death, which potentially can blur a little bit the rapid change, the recent change. Whereas RNA is essentially a copy of the DNA, which is expressed at when organisms live, and it degrades super rapidly. It degrades in hours to days, which potentially is a much better proxy for detecting rapid change. The problem with RNA is that it is very costly. It is hard to manage, it takes time to extract, and it's not commercially viable or for routine testing. Very good news here is that DNA performed equally well as RNA, and this is very, very good news. Another observation that was good is that if we remove those Foraminifera from the total equation, we kept a very strong relationship. It didn't change, which is great. It simplified our workflow and essentially allows to keep only mostly the best indicators. And this was essentially validated with the independent validation set, the third year. To conclude, this was in a world first. In fact, we developed, tested, and validated a multi-trophic Metabarcoding Biotic index, which allowed, which showed really, the reason it was a world first is that it included a lot of samples from multiple years, farms, and region. We demonstrated that the MBI and ES performed equally well, but with the added advantage of the MBI of being able to reduce the result turnaround and time by approximately 2. And also, the DNA performed equally well as RNA, which was a good news for cost-effective and rapid response. Also, very importantly, we were able, through this project, to generate a very valuable new sequence database that we call the Gene Code ID. Essentially, this database now contains all of the signatures that corresponded to these 500 eco-groups that were used to generate the index. And what that means is that future samples will be simply compared directly to this database to generate the index automatically in a click of a button. We consider that the index is ready to go, but any transition like this one needs to be taken with great caution because because it can have a number of repercussions for the environment and for commercial repercussion. So we really, really recommend to perform a phasing period of further ground truthing the tool until 2018. So as a matter of fact, we have hundreds of samples archived in our laboratory since 2012, and we will continue to do so until 2018 in the Marlborough Sound. We have some samples from Stewart Islands, and we are just about possibly to have more from Stewart Islands, and we were lucky enough to have also some samples from a fish farm in Acknowledgments in 2017 this year. So this phasing period would allow us to really push further investigation on better understanding the bacterial and eukaryotic components of the program. It would help us improve the quality and the size of this gene code ID database and test its functionality. It would allow us to test a number of sampling devices that would be particularly adapted for small microscopic organisms, very important. Also, we would be able to apply it to, and we will test the tool to apply it in other environments, such as rocky environments, which is very relevant currently with the aquaculture industry tending to go to high flow sites, which is characterized by more rocky reefs. And finally, also test it in for other marine activities, such as mussel farms or pools. We would welcome the opportunity to engage in discussions with the NTIC Standard Working Group on the uptake of this tool. We thank you very much for your attention. Here are some of the people that really contributed to this project. We thank MDC, particularly Steve, for supporting us all along, and thank you for your attention.
Wonderful. Thanks.